The Echo Code. Imagine you are writing a novel. You finish it, lock it in a drawer, and burn the only copy. 50 million years later, a stranger on the other side of the planet sits down and writes the exact same book. Same plot, same characters, same sentence on page 47. That is not coincidence, that is a message. In 2010, a geneticist named Gareth Jones and his colleagues at Queen Mary University of London published a paper that made biologists go quiet. They had been comparing a single gene across dozens of bat species and toothed whale species. They expected the usual divergence you find between animals separated by nearly 100 million years of evolution. What they found instead stopped the analysis cold. Bats and dolphins had independently mutated the exact same gene down to the same letters of DNA code to build the same impossible superpower. Echolocation. The gene is called Preston. It encodes a motor protein inside the inner ear that vibrates at extraordinary speed, amplifying the faintest returning sound into something the brain can map into a three-dimensional picture of the dark. Bats use it to hunt moths in pitch-black caves. Dolphins use it to chase fish in murky ocean water. The two animals last shared a common ancestor approximately 95 million years ago, when that ancestor was a small land mammal with no echolocating ability whatsoever. They should have built their hearing systems from scratch, independently, using whatever random mutations happened to arise in their separate lineages. Instead, Jones's team found at least 14 identical amino acid substitutions at the same positions in the Preston protein sequence in both lineages. The same changes in the same places in animals that have not shared a common ancestor since the dinosaurs were still alive. The probability of that happening twice by chance is, according to the researchers, effectively zero. But the Preston finding was only the beginning. A follow-up study published in Nature in 2013 by Liu and colleagues identified convergent mutations across more than 200 additional genes associated with hearing in bats and dolphins. It was not one coincidence. It was a systemic parallel reprogramming of the entire auditory system independently executed in two unrelated lineages across two different environments separated by nearly 100 million years. The inner ear structure of echolocating bats and dolphins is geometrically near identical, despite coming from completely different embryological origins. Bat inner ear tissue derives from neural crest cells. Dolphin inner ear tissue develops from the otic placode. Different raw materials, same finished product. The horseshoe bat, and according to some researchers, certain whale species have both independently arrived at a narrow-band echolocation system, a finely tuned, constant frequency pulse used to detect the flutter of a moving target. That is a highly specific computational strategy, one with no obvious reason to appear twice unless something deeper than chance is doing the selecting. Biologists call this molecular convergent evolution. The Preston protein, the argument goes, has only a limited number of configurations that allow it to function as a high-frequency amplifier. Physics and chemistry constrain the solution space. Natural selection, operating under the same environmental pressure in both lineages, keeps arriving at the same answer because that answer is simply the best one available. It is like two engineers on opposite sides of the world independently designing a wheel. Not mysterious, just geometry. But a wheel has one shape because geometry demands it absolutely. The Preston protein is a complex 744 amino acid molecule with enormous configurational space. Identical mutations at 14 specific positions across two lineages separated by 95 million years, even under identical selective pressure, have been calculated as astronomically unlikely to occur by chance. And it is not one gene, it is 200. Some researchers, including evolutionary biologist Simon Conway Morris at Cambridge, have argued that the convergence is too consistent, too molecularly precise, to be explained by the usual story. Conway Morris proposes that life navigates a landscape of attractors, forms, and functions so optimal that evolution keeps finding them almost inevitably. Others go further and ask whether the genome itself contains something like a pre-written library, latent solutions held in reserve, waiting for the right environmental key to unlock them rather than being assembled fresh from random mutation each time. We may never know which explanation is correct, but here is the question that should stay with you. If 200 genes converge independently in two lineages, at what point does the word convergence stop being an explanation and start being a description of something we do not yet understand? The eye that built itself twice is waiting. The eye that built itself twice. Take the most sophisticated optical instrument you can imagine. A variable focus lens, an adjustable aperture, a light-sensitive surface with over 100 million receptor cells, and a neural processing system fast enough to track a moving target in 3 dimensions. Now imagine building it, now destroy it entirely. Now starting from a completely different set of raw materials with no blueprint, no shared ancestry, and no memory of the original design, build the same instrument again. This is what evolution did with the eye. The vertebrate camera eye and the cephalopod camera eye, the eye of the octopus and the eye of the human being, are among the most famous examples of convergent evolution in all of biology. They look nearly identical from the outside. They perform nearly identically under measurement. And they evolved from entirely separate origins, the last common ancestor of vertebrates and cephalopods being a blind, soft-bodied Precambrian worm living roughly 600 million years ago. The strangest part of this story is not the eyes themselves. It is the key that builds them. In 1994, Swiss geneticist Walter Gehring at the University of Basel published a paper in Science that changed developmental biology permanently. He had taken the PAX6 gene from a mouse and inserted it into a fruit fly embryo. PAX6 is the master control switch for eye development in vertebrates. What Gehring found was that the mouse gene transplanted into an organism that last shared a common ancestor with vertebrates over 600 million years ago caused fully functional fly eyes to grow on the fly's legs, wings, and antennae. The mouse gene worked in the fly body. The fly body followed its instructions. The same ignition key started 2 completely different engines and produced, in each case, a working eye. The vertebrate eye and the cephalopod eye are not homologous structures. They are not derived from the same ancestral tissue. The vertebrate retina is what biologists call inverted, with photoreceptors pointing away from incoming light and the optic nerve creating a blind spot where it passes through the retina. The octopus retina is averted, with photoreceptors facing the light directly and no blind spot at all. 2 different solutions to the same optical engineering problem, both controlled at the genetic level by the same master switch. PAX6 has since been found governing eye development in flatworms, box jellyfish, and cubozoans, animals whose eyes range from simple light-sensitive pits to sophisticated multi-lens structures. The gene appears to function not as a blueprint for any specific eye design, but as a gateway, a biological instruction that reads simply, build something that sees. A 2013 paper in Current Biology by Todd Oakley at the University of California, Santa Barbara found that the light-sensing proteins used in octopus and vertebrate eyes, while not chemically identical, both recruit the same molecular scaffold, a 7-transmembrane protein structure, despite independent evolutionary origins. The lens crystallins, the proteins that make the lens transparent, are chemically unrelated between octopus and vertebrate lenses. Different chemistry, same optical properties. The performance converges even when the materials do not. Mainstream evolutionary biology explains this clearly. PAX6 is ancient, versatile, and was already involved in general head patterning long before eyes existed. When selective pressure for vision arose independently in different lineages, PAX6 was the most accessible regulatory lever available. The physics of light capture demands lenses, apertures, and photoreceptors, so natural selection keeps arriving at similar structures regardless of starting material. The convergence is physics forcing biology's hand. But here is what that explanation leaves unaddressed. If evolution is free to build eyes from any available proteins, why does it keep pulling the same lever in organisms that diverged 600 million years ago? Gehring himself argued in a 2011 review that PAX6 may function almost as an unavoidable attractor, a preloaded instruction so deeply embedded in animal genetic architecture that evolution does not create it so much as discover it repeatedly across deep time. Some researchers ask whether the genome contains a latent pattern library, solutions that exist in potential, waiting for the right conditions to be expressed. What built the eye the first time, before any eye existed? That question has no comfortable answer. And the fact that it keeps building the same one from different raw materials 600 million years later suggests the answer may be stranger than we currently allow. From eyes to teeth now, and a killing machine that refuses to stay extinct. The killing tooth. There is a skull buried in the La Brea tar pits of Los Angeles. The upper canines are 8 inches long, curved like a pair of scimitars, serrated along the back edge for slicing through the neck of prey far larger than a modern lion could tackle. It belongs to Smilodon fatalis, the saber-toothed cat, and it went extinct approximately 10,000 years ago. Now, travel south and back in time, deep into the Miocene grasslands of South America, 10 million years before Smilodon stalked the California coast. Dig up another skull. Different continent, different era, different lineage, no cat ancestry whatsoever. And there are the same long, curved, serrated fangs looking back at you from the skull of an animal called Thylacosmilus atrox, a marsupial. A creature more closely related to a kangaroo than to any cat that ever lived. Evolution did not copy the design. It could not have. The two lineages had been separated for over 180 million years. It simply made the same decision twice. In 2020, paleontologist Stefan Lautenschlager at the University of Birmingham published a landmark paper in Royal Society Open Science using three-dimensional digital modeling and finite element analysis to compare skull biomechanics across all known saber-toothed lineages. What he found was not just functional similarity, but quantitative convergence. The stress distributions across the skull during a killing bite, the geometry of the zygomatic arch, the reinforcement patterns in the jaw were near identical between Smilodon and Thylacosmilus, despite the skulls being constructed from different bone architecture, shaped by different developmental programs in animals that had not shared a common ancestor since before the dinosaurs. There are at least 4 completely independent saber-toothed lineages in the fossil record. The Machairodontini, the true saber-toothed cats, including Smilodon, the Nimravidae, the false saber-tooths, which diverged from the cat lineage approximately 50 million years ago. The Barbarophilidae, sometimes called the false saber-tooths, a third entirely separate lineage, and the Thylacosmilidae, the South American marsupials. The Nimravids appeared in the fossil record roughly 37 million years ago, some 15 million years before the true saber-toothed cats arose. Saber teeth evolved, went extinct in one lineage, and were independently re-evolved by a separate lineage in the same geographic region. the solution appeared twice in North America alone. Thylacosmolus atrox had canines that continued growing throughout the animal's life, rooted in bony flanges extending down past the jaw, a completely different mechanical solution to the engineering problem of maintaining an 8-inch tooth. Different architecture, same visible result. Smilodon fatalis canines reached up to 28 centimeters in length. A 2018 study in PLOS 1 identified that the saber-tooth morphology correlates precisely with a specific prey size class, large herbivores over 100 kilograms, suggesting that the environment was issuing the same ecological request to 4 separate evolutionary lineages across hundreds of millions of years. And further back still, in Austranthavia alexandri, a non-mammalian therapsid from the Permian period, approximately 255 million years ago, also exhibited saber-like upper canines. The first independent origin of this tooth form predates the existence of mammals entirely. The mainstream explanation is straightforward and reasonable. Saber teeth are the optimal solution to a specific predatory problem. When prey is large and thick-skinned, a downward stabbing bite to the throat or nape is mechanically superior to any crushing bite. Given the same prey type on multiple continents across multiple geological eras, natural selection keeps building the same tool. It makes sense. But the saber-tooth morphology requires simultaneous modification of the skull, jaw musculature, neck vertebrae, gape angle, and tooth root anatomy, all of which must co-evolve or the system does not function. The probability that 5 separate lineages independently coordinated all of those changes in the same direction without sharing any genetic basis for the trait has not been satisfactorily explained at the developmental level. Some researchers working in craniofacial developmental biology have proposed that large carnivore genomes may carry a latent canine elongation pathway, a dial controlled by a small number of growth factor signals that multiple lineages independently discovered, not building the killing tooth from scratch each time, unlocking it. In the darkness of the deep ocean, another kind of unlocking was happening, and this one lit the whole abyss on fire. The living light. There is a layer of ocean where sunlight never penetrates. It begins at roughly 200 meters below the surface and descends into permanent darkness. No photon from the sun has touched it in billions of years. And yet, if you could hover in that black water, you would not be in the dark. You would be surrounded by cold light, blue, pulsing, flashing, drifting, produced by creatures that have never seen the sun and never needed it. Firefly squid pulsing in coordinated waves. Anglerfish dangling a glowing lure in the void. Dinoflagellates igniting in their millions when disturbed, turning a ship's wake into blue flame. The ability to produce living light has been independently invented by life over 50 separate times. The part that should make you stop is this: the chemistry is almost always the same. In 2015, Stephen Haddock at the Monterey Bay Aquarium Research Institute published a comprehensive analysis in PLOS 1 cataloging the phylogenetic distribution of bioluminescence across the tree of life. He counted at least 40 to 50 independent evolutionary origins and noted something that was, in his words, deeply surprising. The molecular toolkit kept repeating, not because the lineages were related, they were not, but because evolution, reaching independently into the chemistry of life from dozens of different starting points, kept arriving at the same reaction. The reaction works like this. A small organic molecule called luciferin is oxidized by an enzyme called luciferase, releasing the energy of that chemical reaction not as heat but as a photon of light. The process is so efficient it produces almost no waste heat at all, which is why biologists call it cold light. This luciferin-luciferase system appears in radiolarians, jellyfish, crustaceans, squid, and deep-sea fish. Their last common ancestor lived over 700 million years ago, and as far as any evidence suggests, produced no light whatsoever. Yet in multiple lineages, the luciferin molecule itself, the specific organic compound being oxidized, is chemically identical. The same molecule, synthesized independently, used for the same purpose by organisms that have had no biological contact for hundreds of millions of years. A 2019 paper in Science Advances by Opa and colleagues found that cholinterazine, the most widely distributed luciferin in the ocean, is likely dietary in origin in some lineages, consumed from bioluminescent prey and repurposed. Which raises a question with no clean answer. Which came first, the organism that synthesizes the molecule or the organism that eats it? The luciferase enzyme, meanwhile, has evolved independently from at least 7 completely different protein families, fatty acid ligases, aldoketo reductases, and others, each time producing an enzyme that achieves near-identical photon emission efficiency. The wavelength of bioluminescence in deep-sea organisms is almost universally blue, centered between 470 and 490 nanometers. This is not arbitrary. Blue light penetrates seawater more deeply than any other wavelength. Independent lineages, evolving their bioluminescence in isolation, all tune their light to the same frequency, the one that travels farthest in the medium they live in. And on land, in 2020, a paper in Scientific Reports documented that the fungus Panellus stypticus uses a bioluminescence pathway completely distinct from all animal lineages, yet produces light within 10 nanometers of the wavelength used by chemically unrelated deep-sea fish. Biologists explain this through the logic of chemical accessibility. The precursor molecules needed to synthesize luciferin appear across basic metabolism. Bioluminescence, in this view, is a short step from chemistry that already exists, so natural selection keeps discovering it. The blue wavelength bias is physics. The ocean selects it because it works. The convergence is expected, but accessibility does not explain precision. If bioluminescence can be assembled from 7 different enzyme families, why do independent lineages so frequently converge on the same luciferin molecule? Something is pulling them toward the same chemistry across 700 million years of separation. Some researchers have proposed that certain molecules occupy such central positions in the biochemistry of life that they function as hubs, nodes that multiple independent evolutionary paths lead through regardless of starting position. The question some are quietly asking is whether cholinterazine is one of them. A molecule so embedded in the ancient chemistry of life that evolution does not invent it so much as rediscover it over and over in the dark. 6 fish families were asking a different question entirely. Not how to make light, how to make electricity. The living battery. An electric eel can discharge 600 volts, enough to stun a horse, enough to be lethal to a human in the right conditions. It produces this through a biological battery, stacks of modified muscle cells, each generating a small voltage, wired in series like cells in a flashlight, running the length of its body. It is one of the most astonishing adaptations in the animal kingdom. Now forget the electric eel entirely and travel to the muddy river floors of Central Africa. Here lives the electric catfish, Melapterurus electricus, capable of delivering a charge strong enough to incapacitate prey and deter crocodiles. It has no evolutionary connection to the eel. Their last shared ancestor lived approximately 350 million years ago and showed no electrogenic capability. And yet inside the catfish is the same biological battery, built from the same gene, using the same ion channels, producing the same result, not something similar. The same. In June of 2014, Jason Gallant at Michigan State University and an international team of collaborators published a paper in Science that cut through one of biology's most persistent puzzles. They had sequenced and analyzed the genomes and tissue transcriptomes of all 6 independently electrogenic fish lineages and searched for the genetic basis of electric organ evolution. The 6 lineages are the electric eel of South America, the electric ray of the world's oceans, the electric catfish of Central Africa, the elephant fish or mormyrids of African rivers, the ghost knife fish of South American rivers, and the stargazers of coastal ocean floors worldwide. These animals are not close relatives. They are scattered across the vertebrate family tree like 6 separate experiments. And Gallant's team found that, in every single case, the same gene had been independently co-opted to build the electric organ. The gene is called SCN4A. It encodes a voltage-gated sodium ion channel normally found in skeletal muscle cells. In each lineage, the electric organ cells had independently lost their ability to contract, with myosin heavy chain genes being silenced while retaining and massively amplifying ion channel expression. The same functional conversion, the same genetic mechanism, independently executed 6 times across 3 continents and 2 ocean basins. In 2019, a new species of electric eel, Electrophorus volti, was described, capable of discharges reaching 860 volts, the highest bioelectrical output of any known animal. 4 of the 6 electrogenic lineages also independently evolved electroreception, the ability to sense distortions in a self-generated electric field, to navigate and detect prey, a sensory system with no analog in non-electric fish, evolved separately in each case by recruiting the same mechanosensory hair cells from the lateral line system. The electric organ and the sensor for it were independently built twice, 3 times, 4 times, from the same tissue and the same gene. A 2021 study in eLife by Pitchers and colleagues found something even more striking. The regulatory regions controlling SCN4A expression in the electric organ, the genetic switches rather than the gene itself, show parallel modifications in multiple lineages. Convergence at the level of gene regulation is far harder to explain by chance than convergence at the protein level because regulatory sequences are not constrained by the physical requirements of the protein they control. Something was pulling different lineages toward the same genetic switch setting. The mainstream explanation is logical. SCN4A is the most accessible raw material for building an electric organ because it already controls ion flow in muscle cells. Muscle cells are, in a sense, pre-adapted to become electric cells. Natural selection for predation and navigation in murky water drives independent lineages toward the same available solution. The physics and biochemistry of bioelectricity leave limited room for variation. But 6 lineages modifying not just the same gene but the same regulatory switches independently in different oceans and river systems is the genetic equivalent of 6 different authors, working in isolation, writing the same sentence in the same chapter of a book they have never read. Gallant himself described the findings as raising deep questions about the nature of convergent evolution, and specifically whether the genome may contain something like a preformed circuit diagram for electric organ development, latent in all fish, waiting for the right ecological pressure to switch it on. Not 6 inventions, 6 unlockings of a capability that was always partially there. The cold solved its own problem with different chemistry entirely, and it solved it in 2 oceans at once. The antifreeze solution. The blood of a vertebrate freezes at approximately minus 0.9 degrees Celsius. The water of the Southern Ocean sits permanently at minus 1.9 degrees. By every law of basic biology, fish in Antarctic waters should be dead. Frozen solid, their blood crystallizing, their cells bursting from the inside out. They are not dead. They have proteins in their blood that physically grab ice crystals and prevent them from growing, a molecular antifreeze more sophisticated than anything produced by industrial chemistry. Scientists discovered this in the waters around Antarctica and called it one of the most remarkable adaptations in the history of vertebrate life. Then they went to the Arctic and found antifreeze proteins there too. Different fish, different ocean, completely different protein chemistry, doing the exact same job. The comparative work was formalized by C.H. Christina Chung at the University of Illinois in a landmark paper published in PNAS in 2014. Chung and her colleagues demonstrated definitively that the Antarctic and Arctic antifreeze systems had independent evolutionary origins, not just structurally distinct proteins performing a similar function, but proteins derived from entirely different ancestral molecules with no structural homology to each other. The Antarctic antifreeze glycoproteins, or AFGPs, are the more remarkable story. They evolved in the nototheniod fish, a group that dominates Antarctic waters, approximately 5 to 14 million years ago as the Southern Ocean cooled with the formation of the Antarctic Circumpolar Current. Chung's analysis traced their origin to a pancreatic trypsinogen gene, a digestive enzyme. A tripeptide repeat within the trypsinogen sequence was duplicated again and again through a process of gene amplification, sometimes reaching 60 tandem copies in a single genome, one of the most rapid gene amplification events documented in vertebrate evolutionary history. The result was a protein with no structural resemblance to any known antifreeze molecule. It had been built from scratch, from a digestive enzyme, in under 10 million years. The Arctic cod antifreeze proteins belong to an entirely different structural class. Type 1. Antifreeze proteins are simple alpha helices of only 37 amino acids, derived from a completely unrelated protein family. Type 2 antifreeze proteins are structurally related to calcium-dependent lectins, sugar-binding proteins. Neither has any evolutionary connection to the Antarctic AFGP system. Yet both systems inhibit ice crystal growth at temperatures within fractions of a degree of each other, achieving the same thermal protection in two isolated oceans from chemically unrelated starting materials. The ice-binding surface of all known antifreeze proteins, despite being built from unrelated amino acid sequences, presents a flat, regular array of methyl groups to the face of the ice crystal. This geometric feature appears to be a physical requirement of the ice-binding function itself. Whatever protein evolution selects to do this job, it must present this particular surface to the crystal. The chemistry varies, the geometry cannot. And antifreeze proteins have now been independently evolved in insects, including the spruce budworm, in winter ryegrass, and in certain bacteria, each time using yet another structurally unrelated protein architecture, each time arriving at the same ice-binding geometry. The mainstream explanation is compelling. The physics of ice inhibition constrains the solution to a narrow range. Any molecule that will stop ice crystal growth must adsorb onto the crystal face and match its lattice geometry. This physical requirement selects for the same surface architecture regardless of the protein's origin. High selective pressure in a lethal environment plus a constrained physical solution equals convergence. It is expected. But expected convergence on the same geometry does not explain the precision with which two isolated oceans arrived at the same functional performance, the same tissue distribution, and the same protective temperature range in lineages that diverged hundreds of millions of years ago. Chung has described the antifreeze convergence as evidence for what she calls genomic predisposition, the idea that certain precursor genes are structurally and metabolically closer to useful adaptive solutions than others, that genomes carry hidden reservoirs of near-functional sequences that only require minimal modification to become adaptive. In this view, evolution does not invent anti-freeze proteins so much as reveal them from a library of latent near solutions. The Arctic and Antarctic systems are not two inventions. They are two separate discoveries of something that was already nearly there. Deep underground, in the permanent dark, evolution was making a very different discovery, and it was making it the same way, in caves that had never heard of each other. The darkness. Decision. Somewhere in the cave system of Sierra de El Abra in the Mexican state of San Luis Potosí, a fish has been swimming in darkness for 1,000,000 years. It navigates without difficulty, finds food without effort, and breeds successfully in an environment where no light has ever penetrated. It has no eyes, not damaged ones, not vestigial stumps. Just smooth skin where the sockets would be, sealed over as though they were never there. The first biologists who studied these fish assumed this was simple degeneration, the natural decay of an unused structure, the biological equivalent of discarding tools you no longer need. Then they looked at the genetics, and they found that the eyes were not simply rotting away from neglect. Something was actively removing them, and it was always the same thing doing the removing. The key paper came from Yoshiyuki Yamamoto, then at New York University, published in Nature in 2004. Yamamoto's team was studying Astyanax mexicanus, a species with a remarkable property. It exists as two forms, a sighted surface fish and a blind cave form, and the two forms can be crossed in the laboratory to produce hybrids, giving researchers a unique window into exactly which genes control the shift from eyed to eyeless. What Yamamoto found was that in the cave form, a developmental pathway called sonic hedgehog signaling was expressed in an expanded pattern during embryonic development. The sonic hedgehog gene, named SHH, is a master regulator of midline body patterning. In surface fish, it is expressed in one midline domain. In cave fish, it is expressed in two. That expansion of shy activity across the developing head physically suppresses eye tissue while simultaneously expanding jaw size and the number of taste bud receptors. The eye does not simply disappear, it is traded. The developmental resources that would have built eyes are redirected into sensory systems more useful in total darkness. Astyanax mexicanus has independently colonized cave environments in at least 30 separate cave populations across Mexico. In approximately 12 of them, the same expansion mechanism has been independently activated despite the populations being geographically isolated from each other. The same molecular trade-off, executed independently, again and again within the same species in different caves. A 2013 paper in Current Biology by Rohner and colleagues demonstrated that surface fish raised in permanent darkness for only a few generations begin showing early signs of the same SHH-mediated developmental shift, suggesting the genetic machinery for the cave phenotype is not absent from surface populations. It is latent. Waiting. The pattern extends across the animal kingdom. The Olm Proteus anguinus, a cave salamander from the Dinaric Alps of Slovenia, independently lost its eyes after cave colonization estimated at 5 to 15 million years ago, using the same apoptosis signals in the lens and retina documented in cave fish, despite being a tetrapod rather than a fish. The Texas blind salamander, cave-dwelling crustaceans, cave populations of the amphipod Gammarus minus, diving beetles that colonized cave aquifers all show eye reduction paired with olfactory or mechanosensory enhancement in what appears to be a constrained developmental bargain available across the animal kingdom. A 2020 paper in Nature, Ecology, and Evolution by Bilanjaja and colleagues found that eye degeneration in multiple independent cave lineages correlates with upregulation of the same dopamine metabolism pathway, producing increased exploratory foraging behavior as a compensatory adaptation independently in fish, crustaceans, and insects. Evolutionary biologists explain cave eye loss through the logic of metabolic economy. Eyes are expensive. The neural tissue required to process visual information consumes energy disproportionate to its value in a lightless environment. Mutations that reduce investment in the visual system are selectively neutral or mildly beneficial, and genetic drift finishes the job. The SHH mechanism is used because it is developmentally accessible, not because of any deeper directive, but the developmental trade-off is too precise and too coordinated to be entirely passive. The expansion does not simply remove eyes. It executes a specific swap, eyes for jaw and taste buds, that is optimal for cave life. Cliff Tabin at Harvard, who collaborated on subsequent Astyanax research, has described the cave fish system as revealing that evolution has a limited toolkit, that the number of developmental programs available for modifying the vertebrate head is small enough that independent lineages keep arriving at the same program. Others go further and suggest the cave phenotype may be better understood as a developmental release, an alternative body plan held in reserve in the genomes of surface populations, expressed when the relevant trigger is applied. The cave animal, in this view, is not building something new. It is switching programs. Above the surface, in the open air, 4 separate lineages were switching on a different program entirely, one that would take them somewhere no vertebrate had ever been before. The geometry of flight of Boeing 747 and a dragonfly have never been in the same room. One was designed by engineers with degrees in computational fluid dynamics software. The other was designed by nothing, over 350 million years of unguided mutation. And yet, if you measure the cross-sectional curvature of a dragonfly's forewing and compare it to the cross-section of a commercial aircraft wing, you find the same aerodynamic principle operating in both. A cambered surface, thicker at the leading edge and tapering toward the trailing edge, generating lift by accelerating air across the upper surface faster than the lower. This is Bernoulli lift, and it has a mathematically optimal curvature range for any given speed and weight combination. Nature solved powered flight 4 separate times. Every time, it arrived inside the same narrow mathematical window. The 4 origins are genuinely, completely independent. Insect wings are outgrowths of the thoracic exoskeleton, novel structures with no vertebrate homologue. Pterosaur wings were membranes stretched between an enormously elongated fourth finger and the body, supported by a unique tissue called the actinofebrils. Bird wings are modified forelimbs with feathers derived from scales. Bat wings are membranes stretched between elongated hand bones, the second through fifth fingers. The last common ancestor of insects and vertebrates, a Precambrian organism living approximately 600 million years ago, possessed none of these structures. Insect and vertebrate flight are separated by the full depth of animal evolutionary history. A 2017 analysis by Rowan Lingham Solier at the University of KwaZulu-Natal, combined with independent aerodynamic modeling studies, quantified wing geometry across the 4 lineages and found convergent compliance with Bernoulli optimal airfoil curvature ratios in all of them. A 2019 paper in Nature by Desecchi and colleagues found that the upstroke to downstroke power ratio in bird and bat flight is convergently similar, with both lineages independently evolving what is called a power recovery upstroke, a feature not present in their non-flying ancestors and not strictly required for basic flight, but which improves efficiency at high speed. Both made the same efficiency upgrade, independently, after powered flight had already been established. A 2020 paper in eLife by Serrano and colleagues found that the forelimb proportions of early birds and early bats, measured as the ratio of distal to proximal limb elements, independently converge on the same value during the evolution of powered flight, as if the arm geometry of a flapping wing is constrained to a narrow range by aerodynamic requirements that neither lineage could avoid. The pterosaur-bat convergence is perhaps the most striking of all. Both independently chose to elongate their finger bones and stretch a thin membrane between them to build their wings, despite the fact that feathered wings and rigid insect wings are equally viable aerodynamic solutions. Pterosaurs used their fourth finger, bats used their second through fifth. Different fingers, same structural decision. The last common ancestor of pterosaurs and bats was a basal amniot, a small terrestrial animal with entirely normal unelongated digits and no wing membrane. Both lineages, separated by over 165 million years of independent evolution, looked at their hands and made the same choice. The mainstream explanation is that the physics of powered flight is highly constrained. To stay aloft, a flying animal must generate lift exceeding its body weight, which requires a minimum wing area and a minimum airspeed, both of which demand aerodynamically efficient wing shape. The Bernoulli principle operates regardless of wing material, so evolution converges on the same camber geometry. Physics forces biology's hand, and that is all. But physics predicts a range of workable solutions, not a specific structural implementation. The pterosaur-bat convergence on membrane wings supported by elongated digits is not demanded by aerodynamics. It is one workable solution among several. Some evolutionary developmental biologists have proposed that the tetrapod limb developmental program creates a strong bias toward digit elongation as the lowest-cost genetic path to wing construction, because the regulatory machinery for both elongation and membrane development exists in the ancestral limb bud. Physics may select the airfoil shape. Developmental constraint, something preloaded in the genome, may select the structural solution. The sky was not empty before life reached it. In some sense, the blueprint for reaching it may have been there all along. Inside the body, a different and more intimate blueprint was waiting to be discovered, and it was discovered not twice, not 10 times, but over 150. The living womb. The placenta is the most intimate organ in vertebrate biology. It is a living interface between a mother and her developing offspring, allowing nutrients, oxygen, and immune signals to transfer across tissue that the immune system would normally identify as foreign and destroy. It is, most people believe, the defining innovation of placental mammals, the feature that separates us from egg-laying vertebrates and made complex live birth possible. Except that placental mammals are not the only ones. Sharks have placentas, lizards have placentas, multiple species of bony fish have placentas, and scientists who study reproduction keep finding more in lineages that have no business sharing the same reproductive strategy, built from completely unrelated tissue, using independently evolved molecular machinery over 150 separate times. Günter Wagner at Yale University, one of the leading theorists of evolutionary novelty, co-authored a major 2015 paper in Science with Mihaila Pavlichev that examined the molecular signatures of placental evolution across vertebrate lineages and found something that challenged standard explanations at the molecular level. The final product of convergent placentation is striking enough, but it was the implementation that raised the harder questions. Independently evolved placentas in mammals, sharks, and lizards share convergent upregulation of the same immune suppression genes, specifically genes encoding progesterone signaling and uterine natural killer cell regulation, despite these groups having diverged over 450 million years ago. It was not just that they built a placenta, it was that they used the same molecular components to build it. The spiny dogfish shark, Squalus acanthius, develops a yolk sac placenta with direct vascular connections between maternal and fetal tissue, functionally indistinguishable from a mammalian placenta, in an animal whose last common ancestor with mammals was a jawless fish with no uterus. The three-toed skink of Australia, Scyphos equalis, has independently evolved a true chorioallantoic placenta, the same structural type used by humans, from an egg-laying ancestor. More extraordinary still, some populations of Scyphos equalis lay eggs, while other populations of the same species give live birth, making it the only documented case of a vertebrate lineage caught mid-transition between egg-laying and placentation. A 2020 paper in Nature, Ecology and Evolution by Griffith and colleagues examining the genomic basis of Scyphos equalis placentation found that the same calcium transport genes used in human placentation had been independently recruited in the skink, despite no shared placental ancestor in the last 300 million years. The placenta requires solving three simultaneous biological problems: suppressing the maternal immune response to genetically foreign fetal tissue, constructing vascular connections between two separate blood supplies, and regulating nutrient and gas exchange at sufficient efficiency to sustain rapid fetal growth. Each of those problems requires multiple genetic innovations. That vertebrate evolution solved all three independently using convergent gene expression patterns over 150 separate times is one of the least discussed facts in all of evolutionary biology. Wagner has proposed the concept he calls facilitated variation, the idea that certain genomes are biased toward specific evolutionary outcomes because their regulatory architecture makes some phenotypes more accessible than others. The vertebrate genome, in this view, contains what might be described as a latent pregnancy module, a network of interacting genes, each performing useful functions in non-reproductive contexts that can be collectively co-opted into a placentation system with relatively few regulatory changes. The 150 independent origins may not represent 150 separate inventions so much as 150 separate discoveries of the same hidden room in the genome. The mainstream explanation is powerful. The selective benefit of live birth is enormous, providing protected gestation and maternal resource transfer to offspring across environmental variability. Given that benefit, and given that all vertebrates share the basic cellular machinery for immune regulation, vascular development, and hormone signaling, convergent evolution toward placentation is expected. High selective pressure plus accessible molecular tools equals repeated reinvention. But the molecular convergence Wagner identified goes beyond the functional outcome. It extends to the specific regulatory circuits activated, the specific genes co-opted, and in some cases the specific retroviral sequences recruited. If evolution were building placentas from scratch each time, we would expect wide variation in the molecular implementation even when the final product was similar. The implementation converges too. Something in the genome is not being invented 150 times. It is being found. In 100 other lineages, a different room in the genome was being found over and over across 400 million years. And what was in that room was a weapon. The poison library. The cone snail is a beautiful object, a thumb-sized shell resting on a tropical reef, its patterns like brush strokes in porcelain. Pick one up and it will harpoon you with a hollow tooth the diameter of a needle. Inject a cocktail of 200 separate toxins in under a millisecond, and there is no antivenom for many of its most dangerous species. The platypus, a mammal so anatomically improbable that the first European scientists to examine a specimen suspected it was a taxidermy hoax, has a venomous spur on each hind leg that delivers a protein cocktail capable of causing excruciating pain, lasting months in a human adult. The slow loris, a wide-eyed primate from Southeast Asia, secretes toxin from a gland on its elbow, licks it onto its teeth, and delivers a venomous bite capable of causing anaphylactic shock. The short-tailed shrew's saliva is toxic enough to paralyze prey several times its own size. Scientists have now confirmed that venom evolved independently over 100 times across the animal kingdom. And in nearly every case, evolution went to the same library to build it. Brian Fry at the University of Melbourne published a landmark paper in Nature in 2006 that changed the scientific understanding of how venom evolves. Fry and his team analyzed the venom gland gene expression of 24 snake and lizard species and found something that the field had not expected. The venom proteins were not novel inventions. They were ordinary body proteins, phospholipases, serine proteases, three-finger toxins, growth factors, proteins expressed throughout the body in entirely non-toxic functions. The venom gland had not created new chemistry. It had recruited existing proteins, expressed them in a specialized tissue, and then accelerated their evolution under selection for toxicity. The same families of proteins appear in the venoms of snakes, lizards, spiders, scorpions, centipedes, bees, and at least 3 independent scorpion lineages, different animals, different continents, different evolutionary histories, all reaching into the same molecular toolbox. Phospholipase. A2 enzymes appear in the venoms of vipers, cobras, elipid snakes, bees, wasps, centipedes, and multiple scorpion lineages, all recruited independently from the same enzyme family that functions in normal cellular inflammation signaling. The three-finger toxin structural scaffold, the architecture underlying alpha-bungarotoxin, the acetylcholine receptor blocker in cobra venom, also appears in the skin secretions of certain frogs and in the saliva of non-venomous snakes, suggesting the protein scaffold was widely distributed across tetrapods long before any venom system evolved. The platypus venom contains defensin-like peptides derived from immune defense proteins, showing that mammals independently recruited immune system molecules as venom components, the same category of raw material independently co-opted in certain snake lineages. A 2009 paper in PNAS by Whittington and colleagues found that platypus venom defensins had undergone convergent evolution with natriuretic peptides found in snake venoms, different protein families independently modified to achieve the same physiological effect, hypotension, and severe pain in rival males. The cone snail system is built from conotoxins, peptides unrelated in sequence to any vertebrate venom protein. Yet they converge on the same molecular targets, voltage-gated sodium channels, calcium channels, and acetylcholine receptors. Different chemical keys, manufactured in an entirely different organism, made to fit exactly the same locks. A 2014 study in Toxicon identified that a spider and a cone snail had independently evolved peptide toxins that target the same subtype of voltage-gated calcium channel, a specific variant called N-type calcium channel, out of multiple available options. Not the same channel family, the same subtype. Fry has proposed that the venomous condition may be less an evolutionary novelty and more an evolutionary threshold, a state that many lineages approach through normal variation in protein expression and tissue specialization and cross when selective pressure demands. In this view, the genome of virtually every animal contains latent venom potential. The 100-plus independent origins represent not 100 inventions, but 100 unlockings of a capability that was always partially present. The library was always open. The question was which lineage would walk through the door. And that question, repeated 10 times across the items we have covered, is ultimately the same question. Look at what we have just walked through. Bats and dolphins writing the same genetic sentence 200 times over, in the same gene, independently, across 95 million years of separation. Octopus and vertebrate eyes passing the same physics test using different chemistry, controlled by the same ancient master switch. Saber teeth built 4 times from 4 separate starting points with the same skull mechanics in animals more distantly related than you are to a fish. Bioluminescence reinvented 50 times using a molecule that should not be the same across 700 million years of divergence. 6 fish lineages drawing from the same genetic well to wire themselves with electricity down to the same regulatory switches. 2 isolated oceans separated by the entire landmass of the planet arriving at the same molecular antifreeze from completely unrelated protein families. Dozens of cave lineages activating the same developmental swap, eyes for jaw and sensory tissue using the same gene. 4 lineages achieving powered flight through the same aerodynamic window, and 2 of them independently deciding to build their wings from membrane stretched across elongated fingers. 150 species constructing the living womb using the same molecular components, from sharks to lizards to humans, and over 100 venomous lineages reaching into the same protein library to build the same weapons against the same biological targets. The mainstream answer is convergent evolution. Physics, chemistry, and developmental constraint channel life toward a limited number of optimal solutions. The argument is well supported and intellectually honest. It explains the broad shape of what we see, but the mainstream explanation struggles at the molecular level, where the convergence is not just functional, but implementational. Identical amino acids at identical gene positions, the same regulatory switches, the same protein subfamilies co-opted in the same order. In organisms that have had no biological contact for hundreds of millions of years. At some point, the gap between convergent outcomes and convergent molecular implementation is asking for a different kind of explanation. What that explanation might be, some researchers are beginning to sketch. Latent genetic programs, developmental attractors, genomic predisposition, a library of near solutions held in reserve, waiting for the right environmental key. The idea that evolution is less a creator than a curator, endlessly reorganizing the same molecular inventory. These are serious scientific proposals from serious researchers, not the fringe. They are the frontier. And the frontier is the most interesting place to be. Most people hear convergent evolution, nod, and move on. You made it here through the molecular genetics and the fossil beds and the deep ocean and the cave fish and the cone snails. That is not passive consumption. That is the beginning of the kind of thinking that actually advances understanding. This channel exists for people who find the official answer too convenient, who notice that the footnotes contain more truth than the headlines, who understand that the history of science is a history of comfortable certainties being replaced by better questions. Make your membership official. Hit subscribe and lock this place in as your base of operations. Then go to the comments and answer this. Of the 10 patterns we covered, which convergence feels least like coincidence to you? Which one, if you had to defend it in an argument, would be hardest to explain away? Build your case. We will be reading everyone. Life keeps writing the same book in different languages. Eyes that see the same world through chemically unrelated lenses. Teeth that kill the same prey from skulls separated by 100 million years. Light produced by different hands from the same molecular fire. The lesson is not that evolution is predictable. The lesson is that the universe appears to have a very short list of answers to a very large number of questions, and something embedded in the fabric of biology, maybe in the genome, maybe deeper than that, already knows what those answers are. We are just beginning to understand that the randomness was never as random as it looked. The echo code was always in there. Don't stop asking why. Hit subscribe and keep digging.